Okay, so I think I'll just go ahead and get started then with some introductions. So first of all, this is the Mycological Society of America Student and Postdoc Section Colloquium. So I'm Nicole Reynolds, and I'm a postdoc in Dr. Teresa Poloska's lab at Cornell University. And I did my PhD at the University of Florida with Matt Smith. And then I did my undergrad and my master's at Boise State University. And then also that be presenting today is Alicia from Northwest University in South Africa. And so her lab group are actually collaborators with our lab and on this really large, sort of broad scale NSF project that also involves a third lab, Dr. Jesse Euling at the University of Oregon. So basically we've formed this collaborative group with the very, very broad goals of investigating the biodiversity of Mucoromycota fungi and their endosymbiotic bacteria. And so both of us are going to be talking a little bit about our roles in this project and what we've been doing so far. So Alicia is going to be presenting first. And so she did her undergrad at Northwest University as well, and now she's a PhD student there. And yeah, they've been doing a lot of sampling, a lot of hard work down there. So she's going to tell us about what they've been working on. So Alicia?
Thank you. Okay, let me just quickly share my screen with you guys. If you can just enable that, please. Okay, let's see. Okay. Okay, let me just quickly go to the first slide. Okay, there we go. Thank you, Nicole, for the introduction. So yes, I'm Alicia Koch, a PhD student from the Northwest University in South Africa. My main focus at the moment is environmental mycology, and we are working, as Nicole mentioned, on the diversity of Mucoromycota and the drivers that are influencing their distribution and abundance. So as we all know, the fungal kingdom consists of highly diverse eukaryotic species that inhabit a large variety of environments and are responsible for various ecosystem functions. However, today we are looking at a group of early diverging fungi, namely Mucoromycota. Mucoromycota consists out of three subphyla, namely Glomeromycotina, Mortarellomycotina, and Mucoromycotina. Mucoromycota are mainly characterized by their plant association and plant-based ecologies. When looking at the three subphyla, firstly we have Mucoromycotina, and they are classified in three orders, namely Mucorales, Endogonales, and Ampelopsidales. Mucorales are well represented in culture collections and are economically important as the causes of pre- and post-harvest diseases in fruits. They are also known to have an impact on human health, both beneficially and negatively. Next, we have Mortarellomycotina with its sole order, Mortarales. Mortarellomycotina are commonly isolated from soil, and some have been known as root endophytes of plants. However, their effect on the host's fitness is not known yet. Lastly, we have Glomeromycotina that consists of arbuscular mycorrhizal fungi, which, as we know, is the most common form of mycorrhiza. And interesting, the subphylum also consists of Geosiphon, which is an endosymbiont of cyanobacteria. However, the microbiota of plants are considered more complex than merely plant-fungal interactions. The fungal communities associated with plants comprises out of saprotrophic, symbiotic, and pathogenic fungi. While individually, fungi can also form associations with soil bacteria and can also act as a host of endobacteria. Representatives of Mycorrhizata are known to harbor two types of endohyphal bacteria, namely Burkholderia-related endobacteria and Mycoplasma-related endobacteria. The first described endobacteria found in Mycorrhizata was Candidatus glomerobacter gigasporanum. They are known to be transmitted vertically, and they can be found in the mycelium and in the spores of fungi. It has been found that they help the fungus form a relationship with the plant by increasing ATP production and inducing detoxification of reactive oxygen forms. Another similar relationship occurs between Rhizopus microsporus and two species of mycetohabitants. In this relationship, it has been found that the endobacteria completely controls the asexual reproduction and partially controls the sexual reproduction of the host. A more recent Burkholderia-related relationship was found between Leishmania elongata and Mycoavidus. Through their partnership, it has been found that the bacteria rely on the fungal cysteine and that the growth rate of the fungus actually improves if it is cured of their bacterium. Secondly, we have Mycoplasma-related endobacteria. These type of endobacteria were first found in Glomeromycotina, as well as in the genus Mortorella and in the order Endogonales. It is concluded that apart from being endosymbionts, bacteria from the Mycoplasmataceae family can be saprotrophic or mildly parasitic. Okay, when looking at the various relationships between Mycorrhizata and their symbionts, it raises the question as to what the influence of the endobacteria is on the diversity of the Mycorrhizata fungi. And as Nicole mentioned, our lab in South Africa is collaborating with the Paulowska lab at Cornell University, as well as with Dr. Jesse Euling at the Oregon State University to unravel the influence that the endosymbionts might have on the biodiversity of Mycorrhizata fungi. There are three main points to consider, and this includes the functional role in structuring fungal communities across spatial scales, the role in phylogenetic diversification of fungi, as well as the genetic underpinnings of symbiotic innovations. When analyzing the ecological diversity of Mycorrhizata across spatial scales, it is important to consider the dispersal filtering, environmental filtering, as well as biotic filtering. For dispersal filtering, we're investigating whether the fungal community dissimilarity increases over geographic distance. And when we're investigating the environmental filtering, we are looking at indirect gradient analysis. And for biotic filtering, we are comparing Mycorrhizata communities associated with different plant species, as well as the role of endobacteria in the dispersal of Mycorrhizata. To achieve our goal in investigating dispersal filtering, we have selected two mutual ecoregions across the two continents, namely the Mediterranean forest, woodlands, and scrub, and the desert and xeric shrublands. For South Africa, this meant that we had to sample in 4 biomes to cover both these two ecoregions. Our first sampling trip was done at the Wip and Tygerberg nature reserves, in the Mediterranean forest, woodlands, and scrub ecoregion. 3 transects were established. They were 40 meters long with 5 points each, and they were 300 meters apart. So yeah, that was our sampling plan, and we sampled per point. And then our second sampling trip was done at Witstrand and Rolfontein nature reserves. They are found in the desert and xeric shrubland ecoregion. Once again, we had 3 transects. They were 40 meters long with 5 points each, and they were 300 meters apart from each other. Comparing the Mycota communities associated with different plant species meant that we had to look for a dominant plant species that form fungal associations, as well as a co-occurring species. We sampled 3 individual plants per point, which meant that we had six sampling plants per point. The collected samples included rhizosphere soil, root samples, as well as more rhizosphere soil for culturing. At all 4 nature reserves, the dominant species were from the Asteraceae family. Once again, just an overlook of the dominant and co-occurring plants that were found at Rolfontein and Witstrand nature reserves. The dominant plants in this case differed from the ones that we found at the previous 2 nature reserves, but they were still representatives of the Asteraceae family, so they were still comparable with the dominant plant species that we found at the previous nature reserves. For culture-independent tracking, Illumina MySeq sequencing is being performed on the PCR amplicons that we generated from the plant roots and rhizosphere soil, as well as lab fungal cultures. PCR amplification is being done with 2 primer sets selecting for fungi and 1 primer set selecting for endobacteria. Once all the sequence data has been generated, the sequences will then be classified in amplicon sequence variants. Culturing are also being done on rhizosphere soil to obtain pure isolates. This is being done on 10% wheat germ agar, and once pure isolates are found, DNA extractions are performed, which is then followed by PCR amplification, once again with using primers that select for fungi and primers that select for endobacteria, and then Sanger sequencing is being performed on the PCR amplicons. Okay, as all of us might know, arbuscular mycorrhizal fungi are unculturable. So in order to identify the arbuscular mycorrhizal fungi that we found, we have to set up a greenhouse trap culture. This is achieved by adding sterile pool sand that has been autoclaved to a pot and inoculating that soil with sample soil that was collected in the field. We then place 3 maize seeds directly into the soil inoculum, and we water them every other day. Once the seeds germinate, we reap them to make sure that there's only 1 plant per pot left. We continue to water them every other day until they reach the flowering stage, and then they are harvested. And they are harvested, the root system and the rhizosphere that surrounds the root system are also harvested. Once the plants have been harvested, we can continue with the spore extraction. In order to extract the spores, the soil is mixed with water and ran through a series of sieves. You can see on the screen is a more detailed explanation of the method, but basically it just consists out of a series of sieving and centrifuging until all that is left is the AMF spores. These single spores can then be decontaminated, and they can undergo DNA extraction, PCR amplification, and Sanger sequencing. Just to clarify once again, fungal and bacterial primers are being used during the PCR amplification. Okay, so yeah, as you might imagine, this is quite a big project, but luckily we have a lot of support and expertise on the team, and we have some very interesting results, which Nicole will tell you more about. But yeah, our goal is to better understand the factors that are driving the Mycota communities and to see whether the endosymbiotic partners play a role in their distribution and abundance. And through this project, we anticipate to fill the gaps pertaining to Mycota community structure. I would just like to thank the NRF and NSF for the funding that they provided for the project. I also would like to thank every member that is part of the team and my supervisors as well. Thank you for listening, and if you have any questions, you can feel free to ask.
All right, awesome. Are there any questions from anyone? Well, you guys have started some of the greenhouse trap cultures though, correct?
Yes.
But it's not as easy as it looks in the little figures.
Yes, yeah, no, it was a lot easier to draw up the figures than it was to actually keep the plants alive and to maintain growth throughout. Yeah, we did a quick, or not a quick, but we did the first round of greenhouse trap cultures with 30 pots. We were at 30 plants and only 10 of them survived. But luckily we did spore extraction and there was viable spores. So that is still exciting and still good for us.
Yes. Yeah. So in our lab, Lily, who's actually here with us right now, she is an undergrad that has been working in the lab, and she has been working on our greenhouse trap cultures that we did. And unfortunately, you know, our vascular mycorrhizal fungi, they're not the easiest to work with. They're pretty challenging, but we have made some good progress. And so, yeah, we're still working on getting the sequencing results, but so far we have seen quite a pretty good diversity of spores, at least from the samples. So it's great that we're both seeing spores. That's important.
Yes. Did you do the sequencing on the single spores or did you combine them?
Well, so Lily was able to get a few PCR bands from single spores,
Okay.
but the success rate has not been great, even though we've been using Genomify, a whole genome amplification kit.
Okay.
Even then, it still is not working as well as we were hoping. So our next step, actually, we just decided the other day, we're going to try pooling the spores that we get out, and then do Illumina sequencing on the pooled spores.
Okay.
And so, yeah, I'm pretty excited about that, to see how that turns out. So that's what we're working on right now. So
okay.
Let's see. It looks like there's something in chat here. I generally think of Mucoromycota as ubiquitous in the environment. What do we already know about how their diversity changes in the environment?
Shoot, I don't know if you maybe have an answer to that, Nicole.
Well, I think it depends a little bit which group you're thinking about, right? So even though our vascular mycorrhizal fungi are quite challenging to study in a lab setting, there have been a lot of field studies. You know, so since Illumina technology came along, basically, you know, all mycologists have been able to take advantage of that, but particularly for our vascular mycorrhizal fungi, it has been a great tool. And so there have been a lot of these sort of environmental surveys for our vascular mycorrhizal fungi. And there have been a lot of trends found for those fungi. And so in particular, you know, one of the most, I think, interesting findings is, you know, the assumption about our vascular mycorrhizal fungi has always kind of been that most of the species, pretty much all the species, are very general, right? So they don't have very much host specificity. And so the assumption has kind of been that these species of fungi are basically everywhere because they're generalist and they can associate with a variety of hosts. But what these environmental surveys have found is that, yes, there are some generalist species, but there are actually quite a lot more species that appear to be endemic or restricted to certain geographic areas. And so the other part of that too is that the sequencing surveys have discovered a much greater diversity than what was described just based on morphology. And so it appears that our vascular mycorrhizal fungi are quite a bit more diverse than what was previously known as well. So for example, in desert habitats, you see only specific taxa of our vascular mycorrhizae that seem to be found in the desert compared to other habitats and regions, for example. But for other groups, like for the Mucoromycotina and the Mortariella-mycotina, there hasn't been as much focus on them specifically, especially the Mucoromycotina, I would say, because the Mortariella-mycotina, because as Alicia mentioned, many of them are known to be root endophytes. And so they come up a lot in these various environmental sampling studies that people do, because they tend to be in the rhizosphere soils. And so they are commonly detected. And so there is some data like that for that group, but not as much for Mucoromycotina. So because the majority of the species in Mucoromycotina are considered to be saprophytic, and so they don't have a strong association with plants as far as anybody knows. And so the thought there is that, again, sort of like our arbuscular mycorrhizal fungi, the thought is these fungi are super generalist saprophytes, and so they're basically going to be everywhere where there's food sources, right? But that has not been directly tested. And so that's, to me, that's one of the most interesting things that we're going to be investigating for this project, is actually evaluating whether that is the case for that group of fungi or not. That seemed like a really long answer, but I don't know. Hopefully that was helpful. Okay, any other questions about that before we move on? All right. Well, thanks, Alicia. That was a really great overview of the project. And so as you all can see, it's a very large project. It involves hundreds of samples and many different forms of processing in the lab. So it's a really big undertaking. And so it's great that we have such a good group of people working on things. So what I'm going to talk about is a little bit more focused. And so I'm going to be kind of specifically talking about our work that we've been doing on the Mucoromycotina subphylum. And I'm going to be talking a little bit specifically results that we've gotten from one of our sites, which is in the Sonoran Desert in California. So we actually have 4 sites in California, but right now we're just going to be talking about from the Sonoran Desert. And yeah, so I want to start off with a very broad view and kind of talk a little bit more about the diversity of Mucoromycotina, because I feel like they're not really well known by a lot of people. And I feel like there's quite a bit more diversity that exists in the group that people are not really familiar with. So I would like to go over that a little bit and then talk about some of our culture-based results that we've generated so far. So as Alicia mentioned in her presentation, these fungi are what we would call an early diverging group. So this is a phylogeny of essentially all the groups of fungi, right? So up here you can see the Ascomycota and Basidiomycota, which these are what are known as the mushroom forming fungi. So when you think of mushrooms and fungi, these are probably the groups that you're actually thinking of. And so here, all the rest of these other groups are what we would call the early diverging groups. And so the Mucoromycota and the Zupegomycota both are filamentous groups of fungi. And so basically they are microfungi. The vast majority of them never produce any macroscopic structures. And so as you can imagine, that fact alone makes them quite a bit more challenging to study, right? Because you can't just walk out into the woods and see one popping out of the ground, right? You have to use other methods to try to find them. And so the Mucoromycotina, as you can see, it's a subphylum within the Mucoromycota. And it is the one that contains a lot of the diversity that is known in the group. And so I'm going to be giving a few bit more detailed examples about this. But first, I want to also mention that these fungi fairly, over the past couple of decades, they've really become recognized as harboring these endosymbiotic bacteria, which again, Alicia gave a really nice introduction to this about the two different groups that are known, the Burkholderia-related symbionts and the Mycoplasma-related symbionts. And so what you can see for both of these two different groups of bacteria, there is a very strong phylogenetic signal as far as host association. So these bacteria seem to have specific associations with these three different clades of fungi. And so within the Mucoromycotina, the best known example of a bacterial association is with Mycetohabitans and Rhizopus. And so the Rhizopus Mycetohabitans association has kind of become the model for understanding the relationships between these fungi and bacteria. But as you can see, the bacterial relationships are also quite diverse. And what's even more interesting, I think, is that a single fungus can harbor both a Burkholderia symbiont and a Mycoplasma symbiont at the same time. And so there's really pretty much nothing known about those interactions, this sort of tripartite grouping and how that works. So there's a lot of unknowns still in these relationships. Even more recently, there was a study that was published that was using methods very similar to what we've been doing for our project, which is basically growing up Mucoromycotina and screening the fungal cultures for bacteria using PCR. And so that study found a whole nother clade potentially of symbionts. So this para-Burkholderia group here. So what you can see is all these highlighted sequences, these are potentially endosymbiotic bacteria from their fungal cultures. So this could represent a whole new group. The problem is, though, that these results are only based on PCR. And so they really need to be followed up with microscopy, right, to confirm that the bacteria are actually inside the fungal hyphae. But it does provide some initial evidence that there could be a lot more diversity of these symbionts. Okay, so now let's talk a little bit more about the diversity of the fungi. So I think if anybody knows anything about Mucoromycotina, they would probably just think of them as being saprophytes, right? Just out there decomposing stuff. And that is true for many of them, but there are a lot of other ecologies as well. So there are some mycoparasites, so fungi attacking other fungi. There are a couple of species that are associated with arthropods. There are some that are considered plant pathogens, as well as mycorrhizae or root endophytes. And there are also some coprophilic taxa. So these are species that have been found almost exclusively only on dung, which is something I'm particularly interested in and curious about. So let's look at some examples. So probably the one that most people are familiar with is Rhizopus, right? So this is your basic black bread mold is how a lot of people know it. It's very common degrading fruits and vegetables, and you can find it very easily, right? And so it has this very simple, and so a lot of the Mucoromycotina fungi have this sort of very simplistic morphology. So they basically, I kind of describe it as being a ball on a stick, right? So the ball is the sporangium, and then the stick is the sporangiophore, right? And so, and then they have the hyphae connecting up these clusters. So this is probably the most familiar. Mucor as well is another very common one. And as again, you can see it has a little ball and stick morphology. And so, yeah, these are the most, probably some of the most common saprophytes. But if we can talk about a few of the mycoparasitic species, so I only have 3 examples here, but there are actually several more examples in the group. But the ones that I'm showing you here, all of these species are actually parasitic on basidiomycetes. So you can find them on mushrooms out in the forest. So for example, Spinellus here, this one can be found on a variety of basidiomycetes, but it seems to have a preference for mycenas. So these little small, little mushroom guys. And Schizogites also very common on guild mushrooms. But Dicranophora tends to be on boletes. So that, I think, is an interesting differentiation. The other thing is that both Spinellus and Dicranophora tend to prefer to grow at colder temperatures, so they're cycrotolerant, which I think is an interesting ecological characteristic. And then, in fact, Schizogites can be very problematic in commercial mushroom farms. If you get an infection of Schizogites in one of these large growing facilities, it can be a huge problem. But out in nature, you can find mushrooms that look like this. So here is a bolete, and you can see that it's just been basically kind of the spore bearing surface has been taken over by Schizogites. And so you can see these sort of hairy, hairy looking mushrooms out in the forest, and they're infected with Schizogites. For the arthropod associate examples, there are 2 examples in particular. So this one species called Mycotypha microspora is pretty interesting, and this relationship has only been described fairly recently. And what these researchers found was that it seems like this Mycotypha fungus is involved in sort of this tripartite association with this arthropod host, which is a firebrad, and a bacteria, Enterobacter cloacae. And it seems like, from what the experiments that they were able to conduct, it seems like having this combination of this fungus and this bacteria together causes some behavioral changes in the host. And so when these 2 symbionts are present, the hosts tend to have a different sort of aggregating behavior, which I think is a really interesting finding, and there's a lot more that could be done to try to understand what's happening there. And then the other example is Sporodiniella umbellata, which I think is a very beautiful fungus, which you can see here. And so the sporophores have this umbellate pattern, which to me, they look like little flowers. But Sporodiniella is a facultative parasite of many, many different arthropods. But what I think is more interesting is that this fungus can actually also attack the larvae of various insects. But again, very understudied, and it's really not very clear exactly how this association works between the fungus and the arthropod. And then most of the plant pathogens are actually soft rots or wet rots of fruits. So post-harvest pathogens. So Coenifera, Gilbertella, Mucor, these are all things that will attack the fruits mostly. And so as you can see, this can be a really big problem for growers. Particularly Mucor species can survive in cold storage. And so you can imagine if you have this big crop of fruits that get infested with Mucor, it could cause a lot of damage. The only species that I would say that is known to attack the growing parts of the plant is Rhizopus microsporus, which is known to cause rice seedling blight. So it can actually attack the seedlings and kill them. Okay, and so now thinking about the mycorrhizal association in the root endophytes. So the best known group within the Mucora mycotina are the endogonales. And so they actually form structures that are more ectomycorrhizal-like, more similar to ectomycorrhizae. So rather than penetrating into the root cells, instead they form a net-like structure around the outside of the root. And this is one of the few groups of fungi within the Mucora mycota that actually produces macroscopic structures. So here you can see a picture, an example of a little truffle-like fruiting body that is made by one of these endogonales species. And so many of these endogonales can form these little truffle-like structures that you can actually dig up and find if you go truffle hunting. The other set of fungi within this group that are closely related have been termed fine root endophytes. And so these are different from these ectomycorrhizal type. These ones actually form arbuscular-like structures. So they have structures that look very similar to arbuscular mycorrhizal fungi, but they're in a completely different group phylogenetically. They're not closely related to arbuscular mycorrhizal fungi. And so this image up here is showing a micrograph of a plant root, and all these blue structures are the fungal hyphae. And so here you can see one of these arbuscule-like structures being formed. And so this group has actually the fine root endophytes have only really been described relatively recently, and more and more diversity has been found using these sort of sequence-based tools. A couple other examples, Umbellapis species have been also detected as root endophytes. And they actually have been some experiments that have found that orchids specifically, when they're co-inoculated with Umbellapis and ectomycorrhizae, it improved the growth of orchids. Another relatively recently described group is the Pygmaeomycetaceae. And so this is, these fungi were discovered not far from here in New Jersey, and they were found associated with the roots of mountain laurel and pitch pine in very acidic soils in this particular habitat in New Jersey. And then the last example is Bifiguratus adelaide. And so here's some culture images of both Bifiguratus and Pygmaeomyces. And so you can see they don't have the typical growth form that you would expect from a fungus in culture. It has a very different sort of appearance in culture. But still, since these groups were relatively recently described, these two, there's still quite a lot of work to understand what their role is in the ecosystem. And finally, I think one of the major aspects of these fungi is that many of them have the ability to be opportunistic human pathogens. And so they cause very severe disease known as mucormycosis in humans. And so if you're curious and you Google mucormycosis, just be careful, because there are a lot of really disgusting images of cutaneous infections caused by these fungi. So there are a couple different routes of infection. The most common route, I would say, is probably inhalation of the spores. But as I mentioned, they also can sometimes get in through abrasions in the skin. And so one of the most important features that enables a fungus to be a pathogen in humans is thermotolerance, right? These organisms have to be able to survive our high body temperature. And so one of the most common causal agent of mucormycosis are Rhizopus species. And so as you can see in this figure here, there is a range of thermotolerance across these different Rhizopus species, with Rhizopus microspores and Rhizopus, what they're calling Caspiotus. They have a huge range all the way up to 50 degrees. But as you can also notice, interestingly, these close relatives, Schizogites, remember that's the mushroom parasite, and Sporidiniella, which is the arthropod parasite, they both have very low temperature range tolerance. And so I think this is in particular is really interesting to look at these sort of relationships and try to understand how thermotolerance affects the ecology of the fungi. But as you can see, there are many other species that are capable of being pathogenic as well. And so this issue of thermotolerance, I think, makes the idea of sampling in these really extreme desert habitats particularly interesting. And so that brings us back to our project and the Sonoran Desert. So here's me and the other postdoc that was on the project at the time, Kevin, Rashid, the PI from South Africa, and then Teresa, my PI. And so here we are at this site in the Sonoran Desert doing our sampling. And so similar to what was the methods used in South Africa, we have our transects. We had 3 of them that we set up, and then we were sampling from the roots of 2 different host plants here. And so these 2 different plants, the Laria tridentata and Ambrosia dumosa, are very common across the site and co-occurring along our transects. And so we sampled from 2 representatives of these host plants, or I'm sorry, 3 individuals of these plants at each point along the transect. And so all of these soils, as Alicia mentioned, we did Illumina amplicon sequencing, and we've done culture-based analyses, and then also the trap cultures for AMF. But I'm just going to be focusing on our culture results that we've gotten. And so the main bulk of this work was actually the culturing, and the PCR was actually done by an undergrad that I was working with in the lab, Jack. And so he was in the lab with me for about a year, 2 semesters, I would say. And he did a lot of work. Basically, what you do when you're looking for these fungi, you take the soil sample that you've collected from the field, and you just sprinkle a small amount of it on an agar plate, as you can see here. And then basically you just watch and see what grows out of it. And once you see your fungi of interest starting to grow, then you can use sterile technique to try to isolate those particular species and get them into pure culture. And so once we have a pure culture, then we can use PCR to, first of all, identify the fungus, and then secondly, screen the fungus for potential symbiotic bacteria. So from just this one site, we were able to get about 108 cultures. And from the PCR screening, we found that at least 18 of those had some kind of bacteria sequence, 7 of which came from what are known bacterial symbionts. And so the rest are other types of bacteria that have not yet been characterized as being known symbionts. And of course, the culture-based results recovered much less diversity than the sequencing results, as would be expected. And so about 99% of all of the samples in culturing had some kind of Rhizopus. And so Rhizopus was by far the dominant species we got from cultures. The next most common was Actinomucor. We also got we did get a few mucors and Cunningham and a couple other things, but these 2 were definitely the most common things we found. And so here I'm showing you a phylogeny just of Rhizopus, including sequences from all our samples. And so these names that start with a number, those are all our samples. And so what you can see in this really large clade right here, the majority of the isolates that we got fall within this Rhizopus oryzis clade. And so here in this image over here, you can see some nice micrographs of Rhizopus oryzis. And one of the main things you can notice is how dark it is. It's very, very melanized. And so when you consider the habitat where these fungi are living, this really harsh, UV saturated, desiccated environment, I think having that melanization is probably a key factor to helping them survive. And then what you can also see is that several of these isolates had different kinds of bacteria detected from them. So in the green, these are sequences of bacteria that came back as being called mycetohabitans, which, as I mentioned earlier, that's the known symbiont, right? But then these ones in orange had different sequences of bacteria, which may or may not be actual endosymbionts. And then down here in this other group, this is the Rhizopus microsporus group. Again, also very dark, very melanized hyphae and sporangia. But here you can see, again, many of these isolates had mycetohabitans. And so Rhizopus microsporus, of course, is the one that is known to have mycetohabitans. But what you can also see is that there's a lot of diversity in the fungus Rhizopus microsporus, because what we have is it's actually all these different sequences are differentiating out into these different clades. And so potentially some of these isolates might represent a new species of Rhizopus, but that requires a little bit more work phylogenetically and morphologically to understand that. Okay, and now we are looking at a phylogeny of the Actinomucor species and isolates that we've recovered. So in this blue box, these are all our cultures that we got from our samples. And so what you can pretty clearly see is that they are forming a completely separate clade from all the other known Actinomucor species. And so this is very strong evidence that we most definitely have a new species of Actinomucor. And so this image over here is showing some nice micrographs of what the type species Actinomucor elegans looks like. So you can see it has this sort of branched morphology with these little egg-shaped columella. But basically what I find really interesting about this is that all the known species of Actinomucor elegans are human pathogens. And this particular species here, Actinomucor elegans variety Kuwaitiensis, this was described because it was actually isolated from an infected wounds of soldiers that were fighting in the Middle East. And so several of these species are known to be from these really dry desert habitats. And so I'm really interested to know, our species, which is probably a new species, is it also capable of being a pathogen? Particularly because this Sonoran Desert site that we sampled from is literally about a mile outside of the Palm Desert City in California. And so you can imagine if you have this reservoir of a potential human pathogen, that could be an important thing to know. Okay, so now what I'm showing you is a really big tree of 28S sequences of representative fungal sequences from GenBank, and then included with our data that we got from our project from this site. So in yellow are the OTUs that were generated from our Illumina sequencing data. And so what you can see is that there's a lot of diversity, actually. You can see these yellow OTUs coming out in many different clades all across the tree. So quite a good diversity of Mucoromycotina just from this one site. In red are the sequences from our fungal cultures. The green are the fungal cultures that we grew that also had bacteria detected in them. And so you can see the main group of fungi that had bacteria is Rhizopus, but also we had a few actinomucor that were had bacterial sequences associated with them. And so now, if we look at the phylogeny of the bacteria sequences that we got, again, you see here are our bacteria that we found from Rhizopus. So many of them are coming out in these known Mycetohabitans clades. But what you can also see is that there are several more that come out in different clades. And so over here, for example, these sequences that we got from our cultures are intermixed with these Paraburkholderia. So if you remember that study that I mentioned back at the beginning, found all these Paraburkholderia sequences, some of ours come out very close to those. And then we have a variety of other sequences here that are close to this bacteria called Cooper avidus. And many of these species of bacteria are known as nitrogen fixation capability. So this is potentially a very interesting relationship to look at. So essentially, we found potentially some new species of fungi and potentially new bacterial fungal associations. And so now the next steps, of course, would be to try to follow up these associations with microscopy to really confirm, are these bacteria actually endosymbionts? We also are really interested in understanding what is the frequency of this endosymbiont in these natural populations, because as you can see from our results, only a proportion of the fungi, a small proportion really, have symbionts. And so why is that? Why do we see these patterns? You know, what is driving that? And then finally, as I mentioned, you know, these desert sites have, I think, a great possibility of actually being a reservoir and an evolutionary sort of proving ground for opportunistic human pathogens. So I'm a big fan of the desert, and I think there's a lot of really interesting findings to gather out there. So with that, I'll just thank all our collaborators, and thank you all especially for being here and listening to our research. So if you guys have any questions, just let me know. Alrighty. Well, oh wait, let's see. Might be something in chat. Let's see. So do you use a medium that specifically selects for mucormycota, or do you just throw out all non-mucormycetes? That's a good question. Yeah, so there's not really a medium that's specific for Mucormycotina, I would say, but we do use a very low nutrient recipe. So we use a recipe called wheat germ 10. So it's 1/10 strength wheat germ agar. So it's very, very weak, medium. And then another thing that really helps for isolating Mucormycotina is if you add a selective fungicide that's called Benamil. So Benamil is a fungicide that particularly inhibits a lot of ascomycetes. And so we definitely get an increase in Mucormycotina fungi when we add that to the medium. But other than that, yeah, there's not much that can be selective because they are very generalist saprophytes. And so they can grow on a variety of different things, just like other saprophytes. So basically, when you're looking at these plates from soil, you see a bajillion different things, right? And so you, it really takes some careful microscopy and multiple replicates, if you can, to find these fungi that we're looking for. Here's another question. I'm re-looking at some RNA-seq data from a contaminated sample and have found a lot of bacteriophages. Have you found any viruses in your work? So I have not come across any virus sequences in our data yet. So the primers that we've been using they're meant to be fungal specific. So we're using several different primer sets, I should say. 2 different primers for fungi. So we have a set of 28S primers, and we're also doing ITS primers. And the 28S primers are much less fungal specific. So we do get a lot of off-target amplification. We get a lot of other eukaryotes, but I have not seen any virus sequences yet. But that's interesting though.
Just to add on to that, if there's any curiosity. So a couple of the commonly studied Rhizopus microsporus strains harbor a couple of narnoviruses that have been looked at by Lila Partida Martinez's group. And actually there's some interesting interactions between hosting both narnovirus and endobacteria. And so if that's of interest, it's pretty cool work.
Oh, yeah. Yeah, right. That was a really cool study. And it's really kind of amazing. So this study Meg is talking about, they were somehow able to they created a cured strain of the fungus where they were able to get rid of both the bacteria and the virus, right? Isn't that right, Meg?
Yes. Yeah. And yeah, they saw differences based on whether they were cured of like both virus and bacteria or one or the other. It was pretty interesting. And there's a couple, I think a single fungal host hosted, I think 2 different narnoviruses in addition to the bacteria. So potentially some interesting kind of 3-way interactions between not only fungi and bacteria, but viruses as well. So.
Oh, yeah. No, it's super interesting. And actually, you know, mycoviruses in general are really understudied, but particularly as with most things in the early diverging fungi in our groups. So there has been, so I think if I recall, Tim James was doing some work on microviruses, his lab. They were looking at mycoviruses, particularly using like large scale sequencing surveys among early diverging fungi specifically. And so there's a couple other groups I think that are looking at that, but they're really understudied and potentially really fascinating associations happening there. So a lot of work to be done. Okay, any other questions or comments or anything? All right, great. Well, thanks everyone so much for joining us. And this has been recorded, so I think we'll be posting it somewhere. So later on, if people that were not able to be here, let them know that there's a recording and they can come back and watch it and find out about our project. Okay, thank you everyone.